My talk today is entitled Genetic Parameters for VIA-derived muscle, bone and fat weights in Texel-sired crossbred lambs. This work was led by INRAE and Tolkamp, but I'll present it today, and it was from a project in collaboration with the Texel Sheep Society and ABP Food Group. In the UK, the Texel is a predominant terminal sire breed. So there are about 13 million breeding ewes in the UK, and in the last census in 2012, 27% of them were mated to a Texel ram. Genetic improvement in the Texel breed is therefore very important because it has a significant impact on the UK sheep industry. However, most of the genetic selection has occurred in purebred Texels, whereas the main slaughter generation are crossbred, often with a Texel sire. So therefore, there could be some disconnect between the genetic selection that's happening in the pure terminal sire breed and the crossbred progeny performance and carcass data that are being produced from the slaughter generation. Of interest to terminal sire breeders and lamb producers is the recording of carcass traits. But generally carcass traits are quite difficult to measure. There's a limited amount of recording against individual animal identification. And this has been linked in the past to the fact that traceability through the abattoirs and cutting plants is difficult. However, now that all lambs in the UK have to have electronic identification tags, this makes the retrieval of carcass information more accessible. And the kinds of carcass traits that would be of high value to obtain are information on meat yield and tissue distribution. So this would be information that's valuable for processors and therefore for breeders. Although these are hard to measure traits and haven't routinely been recorded in the past, there's new potential with the likes of video image analysis technology that can be set up in abattoirs. And this kind of technology can feed back information on carcass composition and potentially composition of primal regions within the carcass. However, VIA technology is not at the moment routinely used in sheep abattoirs in the UK. However, as part of the project that I'm describing results from today, a VIA system was installed in an abattoir in England belonging to ABP. And as part of that research project, we collected data on UK slaughter lambs and analysed them, and some of those results I'll present today. So the aims of this talk are to estimate genetic and phenotypic properties of growth and VIA-derived carcass traits in Texel-sired crossbred lambs. The traits that we want to concentrate on are weights of muscle, fat and bone in the carcass and also in individual primal regions, those being the shoulder, the saddle and the hind leg regions. And the aim of estimating these parameters is to inform future genetic improvement for these traits, including information from crossbred lambs. The first step in the project was to calibrate the video image analysis machine against another measure of carcass composition, and we used CT scanning as that gold standard measurement. So in the calibration trial at the beginning of the project, we selected 252 Texel cross Clelland lambs. We put the carcasses through the VIA machine at ABP, and then we CT scanned the carcasses after they'd been through the VIA machine. And we selected these carcasses from across the Europe grid, so with a range of conformation and fat grades to reflect the typical distribution within the UK slaughter population. And the technology provider, E+V, who had provided the VIA machine, then took this data from the CT scanning and related it to the VIA parameters to produce prediction equations for the traits that we were interested in. So these prediction equations were then validated on a separate subset of the data. And high accuracies were produced, particularly for muscle and total primal weights, but also for fat weights and bone weights. So the table at the bottom shows the traits that we were interested in. So in terms of the regions of the carcass, we were interested in looking at the total carcass, the hind primal, the mid primal, and the fore primal. And the separation between these primal regions was between the 6th and 7th lumbar vertebra and the 5th and 6th thoracic vertebra. So that split the carcass into 3 sections. And then within each of those sections, we estimated muscle weight, fat weight, bone weight, and total weight. So these are our VIA-derived carcass traits that we were interested in. So for the larger project, we collected data on farm and from the abattoir across a number of phenotype farms. So in total, we had 2,340 Texel cross Clelland lambs that were produced. And we followed both male and female lambs across 2 years. So they were born in 2018 and 2019. So the data came from 6 farms across the UK that were part of the Texel Sheep Society's phenotype farm network. And in the map on the top here, you can see these farms highlighted with blue dots. So they were spread across the UK. The green and red dots show where the sires came from that were used on the project. So the on-farm data that was recorded included weight data at 8 weeks and at 16 weeks of age when the lambs were weaned around 16 weeks. And then we also calculated a slaughter age for each lamb. And then once they went to the abattoir, we collected more information based on these VIA traits that I showed in the last slide. So looking at weights of fat, muscle and bone in the total carcass and in the 3 primal regions. So all these measurements were taken at a single abattoir in England that belonged to ABP. We then combined the on-farm and slaughter data with pedigree information from the Texel Sheep Society. And the pedigree file that we used included information from over 16,000 individuals. So we had the 2,340 crossbred lambs from the project who had come from 75 different Texel sires and over 1,200 Clelland dams. And we verified their parentage by genotyping as part of the larger project. There was no pedigree available on the Clelland dams on the maternal side of the pedigree, so we just used a sire model in our analysis. For the statistical analysis, we used ASREML, and we had different models for each group of traits. So for the VIA carcass traits, we adjusted for carcass weight, as well as relevant fixed effects of year, farm, sex and slaughter batch, of which there were 54 slaughter batches across the 2 years. And then we fitted random effects of sire and a permanent environmental effect of dam. For the growth traits, the lamb weight traits, we fitted lamb age as a covariate, fixed effects of year, farm, sex, dam age and birth rank, and the same random effects. And for age at slaughter, we fitted year, farm, sex, and the same random effects. So the heritabilities for these traits can be seen in the 2 tables here. The top one is the VIA carcass traits, and the bottom one is the growth traits. All the traits that we looked at in the crossbred lambs had low to moderate heritabilities. However, there was less genetic control of the traits that focused on the mid-region primal. But in general, it seems there's scope for genetic selection for these crossbred growth and carcass traits, and some of them have quite promising heritabilities. Looking further at the results from the genetic analysis, I just wanted to pick out some examples of the estimated breeding values, or EBVs, that were produced. So the top graph shows muscle weight in the whole carcass the EBVs for each of the project sires. So there were 75 sires on the project, and their EBVs ranged from -163 grams to +201 grams of muscle weight. So the difference between the highest and lowest EBV was 363 grams of muscle weight, which is approximately 3% of the average muscle weight of the lambs on the project. Similarly, we could look at the EBVs for the muscle weights in each of the 3 separate primal regions, and I've combined them on the bottom graph here. So we can see that some sires had higher EBVs for one region than another region. And again, the range of EBVs was about 2 to 5% of the total muscle weight within these regions. So in conclusion, there is potential for VIA technology to enable genetic selection for hard-to-measure carcass traits. Within this project, we've developed models, genetic parameters, and processes which could be used in future evaluations by the Texel Sheep Society to incorporate crossbred data into their genetic selection programme. Continuing to record crossbred lambs will help to build this data set, increase genetic linkages, and increase accuracies of breeding values that are produced. And there are plans by the Texel Sheep Society and ABP as a breed society and the processor to exploit these results going forward. And presently they're looking to develop a pilot commercial progeny test producer group to exploit the results in future. I'd like to thank you for listening and thank the funders and contributors to our project.